This is 806 in the protein synthesis chapter. And what we want to do in this is we want to talk about the difference between mRNA and tRNA. And we said back in the intro chapter that mRNA stands for messenger RNA. Messenger RNA. And that tRNA stands for transfer RNA. Transfer RNA. And they look very different. So what we have, and we show it attached to the ribosome, and we're going to see in the next video how this actually works. But each one of these is an individual nucleotide. So this is the mRNA. And what we see here, by the way, I'm going to try to keep the mRNA in green and the tRNA in blue. These are the nucleotides of mRNA that we saw that they were assembled back in the nucleus. So this was put together back in the nucleus off of the, whoops, ASSEMBLED. So this was assembled in the nucleus from the DNA, and I'm going to use a word here called template. So a template is a pattern or something that gives us an order. And so when that DNA opened up, it's considered to be a template. And we build on, so like you're going to do a, you're going to carve a pumpkin, you put the template pattern on the pumpkin. So a template is something that you build off of. And we built this strand of mRNA from a strand of DNA back in the nucleus. It has left the nucleus, come out to the cytoplasm. So all of this now is in the cytoplasm. And we could see that the mRNA assembles into a strand. The tRNA doesn't do this. So the tRNA, first of all, it is in individual tRNA molecule pieces. So let's talk about tRNA, the characteristics. It has individual pieces. And it never is more than two hooked together at once, with a max of 2 that are together at once. It never assembles into a strand. We never have a long strand of tRNA. And the reason is what it does. So here we go. Let's look at the structure. Now the actual structure, I'll kind of just draw it over here to the side. It looks a little bit like a 4-leaf clover. Let's see if I can kind of. So the part that's in yellow here, this box that's in yellow actually looks like this. And on one side, it has 3 nucleotides. So this would be the A, G. And on the other side, it has an amino acid. AA for an amino acid. So the other characteristic of tRNA is that it has 3 nucleotides on one side. On one side. We call this an anticodon. An anticodon, because it's the opposite of the codon. So if you look down here, do you see that this piece of mRNA says AAA, and this is my codon? So the anticodon to that is UUU. On the other side of the tRNA is an amino acid. So the opposite side holds an amino acid. Which is the monomer of proteins. So this is what we're going to use to build proteins. And what we're going to see is the reason. So how do we come up with that table? How do we know that AAA is always going to produce a lysine? This is why. If we look at our mRNA, here I have a codon, AAA. The anticodon is UUU on a tRNA. Every tRNA that has a UUU has a lysine attached. Only lysine can attach to a tRNA molecule that has UUU. Only, this is aspartic acid, only aspartic acid can attach to a tRNA that has CUA. Only a PHE or phenylalanine can attach to a tRNA that has AAG. So we have specific binding between an anticodon and an amino acid. So this is really important. So I'm going to switch to red and all caps. There is specific bonding or attachment, specific bonding for anticodons and amino acids. Scientists figured this out by sticking a tRNA atom or molecule of tRNA into a dish with all of the amino acids and then analyzing and seeing which amino acid was gone. So they took a tRNA that they knew had AAG in it, and they tossed it into this big dish of amino acids. And when they analyzed it, all the phenylalanine was gone. And then they took a tRNA molecule that had a UUU on it. They tossed it into a dish. And when they looked at the amino acids afterwards, all the lysine had been attached and it was used up. So this is how that table is manufactured. We know that AAG will always be phenylalanine. These are the anticodons. The table is read as codons. So let me just add that too. Table is read in codons, and that's in all caps. That's why I went over it in the last video when we did transcription. So what are our takeaways? mRNA and tRNA are very different animals. mRNA are individual nucleotides. It gets assembled in strands. So let's add that too. It is exists as one long strand. tRNA has this unique shape. Again, they show it as a little block. It's actually a clover. One side has 3 nucleotides called the anticodon. The other side has an amino acid, and it's specific. This anticodon can only contain phenylalanine. This one can only contain aspartic acid. This one's lysine, so on and so forth. The ACC was tryptophan. And what we're going to see is your body uses this like a code to build proteins. If that doesn't kind of blow your mind a little bit, I don't know what will, because it is a super, super cool concept. It's like you've got every ribosome in your cell is a secret decoder. If you've ever had a decoder ring or a game where you had to do decoding, that's what happens. And we go from, what we're going to see is we go from our nucleic acids here to our proteins. So we've been saying all year long, DNA is the, I'm waving my hands, can you see the shadow? It's that important. We said DNA is the plan to build proteins. This is where it actually happens. This is where we go from a nucleic acid to a protein. And it's all because of the interface between mRNA and tRNA. So in the next video, we're going to look at the specifics of this and how this actually comes about. All right. Next video is translation. Thanks for watching.